Hello everyone, I am Aruna Punna, working as junior lecturer in botany in Government Junior College for Girls, Bongir. In this class, I am going to teach you about the molecular basis of inheritance with the salient features of DNA double helix structure, central dogma in molecular biology, how to calculate DNA length and number of base pairs, packaging of DNA in prokaryotes and eukaryotes, nucleosome structure, non-histone chromosomal proteins, euchromatin and heterochromatin. Salient features of DNA, double helix structure. Almost I gave more information about DNA, but once simply we discuss the salient features of DNA. In DNA, two strands are present. These are the two strands. So, it is made up of two polynucleotide chains. And these chains are arranged in anti-parallel polarity. So, here, this is one chain. This chain end is 5' end, and this chain continues like this, and other end is 3' end. Automatically, here, if the chain has 3' end, another chain has 5' end. So, 5' end continues like this and end with 3' end. So 5' end and 3' end, they are arranged anti-parallel in direction, then only between two polynucleotide chains, hydrogen bonds formed. And sugar phosphate backbone. This strand is completely made up of sugar and phosphate. So that is why this is called sugar phosphate backbone. This is only main out framework of the DNA, and the nitrogen bases project inside of the backbone. So, to this sugar phosphate backbone, nitrogen bases are fixed inside, project inside. So, here nitrogen bases are purines and pyrimidines, adenine and guanine are purines, thymine and cytosine are pyrimidines. Purines always bind with pyrimidines, adenine always bind with thymine. Between adenine and thymine, two hydrogen bonds are present. Between guanine and cytosine, three hydrogen bonds are present. So here adenine, thymine, two hydrogen bonds, guanine and cytosine, three hydrogen bonds. If here towards this strand cytosine is present, means automatically other side guanine is present. Here T means here A is present. In this way, it continues. And the distance between two strands, 2 nanometers or 20 angstroms. So between this and this, between these two strands, the distance is 2 nanometers or 20 angstroms. That means 1 nanometer is equal to 10 angstroms. What is nanometer? Nanometer is equal to 1 billionth of meter or 10 to the power of minus 9. I will explain this nanometer by taking this normal measuring scale. So in your stationery box, you have common measuring scale which is in centimeters. Here, this measuring scale divided into 10 centimeters. Each centimeter divided into 10 millimeter. Millimeter, you know about this measurement. If this 1 millimeter divided into 10 lakhs times equally, that small portion called as nanometer. Here, two chains are coiled in right-handed fashion. Already I taught you. Here, these two strands always coil towards right-handed side. And the pitch of helix. Pitch of helix means one complete helix. One complete helix means, see here, from here to here. So here this coil circled in this way and completed here. So from this region to this region, this is complete cycle, complete coil. In this coil, in this circle, how many nitrogen bases are present means 10 nitrogen bases are present. So 10 nitrogen pair, BP, base pairs are present. This is one base pair. This is 2, 3, 4. Here one is hidden. We are unable to see. 5, 6, 7, 8, 9 and 10. This is also one is hidden in the coiling. So in complete cycle, complete helix, how many nitrogen base pairs are present means 10 base pairs are present. The length of the DNA is 3.4 nanometers. The length of complete pitch is 3.4 nanometers, and how many nitrogen base pairs are present means 10 nitrogen base pairs are present. Between each successive base pairs, the distance is 0.34 nanometers. So between two successive base pairs, the distance is 0.34 nanometers, and complete 10 base pairs, the total distance is 3.4 nanometers. How a double helical structure of DNA show lot of stability? Because of two reasons. One is these nitrogen base pairs stalks are arranged completely parallel, that means completely horizontal, one above the other. Due to this reason, it shows lot of stability, and one more, due to presence of hydrogen bonds. That is why always the proper 2 nanometers distance is maintained. After the discovery of DNA, very soon Francis Crick proposed the central dogma theory in molecular biology. According to this theory, genetic information flows from DNA to RNA, RNA to protein. Most of the living organisms have DNA as their genetic material. In those living organisms, the DNA undergo replication and this DNA by the process of transcription produce mRNA. mRNA means messenger RNA. In RNAs, 3 types of RNAs are present: mRNA, tRNA, rRNA. About those RNAs I will explain later. And this mRNA translate its genetic information into protein. Whenever protein synthesized, the character is expressed. In this way, the genetic information which is present on DNA transcribed to mRNA and mRNA genetic information translated into protein. In this way, finally, as a protein, the character is expressed. In some retroviruses contain RNA as their genetic material. Example, HIV, human immunodeficiency virus. In coronavirus also, RNA is present as their genetic material. In these retroviruses, the flow of information is in the reverse direction. How it is? Here, HIV virus is having RNA as their genetic material. Now, what happens? By the process of reverse transcription, DNA is produced from RNA. Again, DNA undergo replication, that means multiply so many times, and this DNA by the process of transcription produce mRNA, messenger RNA. So the genetic information is passed from DNA to mRNA. That genetic information which is present on mRNA translated into protein. Whenever protein synthesized in the living organism, character is expressed. Here, what is the difference between transcription and reverse transcription? Transcription means mRNA produced from DNA. Reverse transcription means DNA is produced from RNA. In this way, the flow of information is in the reverse direction in the case of retroviruses which are having RNA as their genetic material. When we see the packaging of DNA helix, how a lengthy DNA condense and form chromosomes which are located in a particular region, that is nucleus. Here, one interesting calculation. Calculate the length of DNA double helix in a typical mammalian cell. Here, mammalian cell is human cell. The total number of base pairs in human cell is 6.6 into 10 to the power of 9. The distance between two consecutive base pairs, you know all, that is 0.34 nanometers. When this 0.34 nanometers converted into meters, that is 0.34 into 10 to the power of minus 9, because nanometer is equal to 1 billionth meter. So here the answer is, when we calculate this problem, the distance of one base pair is equal to 0.34 into 10 to the power of minus 9 meters. How many base pairs are present in one single cell? 6.6 into 10 to the power of 9 base pairs. So then what is the length? So 6.6 into 10 to the power of 9 is divided by 1 into 0.34 into 10 to the power of minus 9. The answer is 2.244 meters. Actually, the human cell nucleus diameter is 10 to the power of minus 6 meters. That means 1 millimeter if divided into 1000 times, that each equal part is equal to 1 micrometer. But here this 2.244 lengthy DNA chromatin shows so many coilings and finally condensed into chromosomes. All these chromosomes located in this very small minute nucleus. In this way, just you can imagine how DNA helix packaging is very complex. And another interesting calculation, if the length of Escherichia coli bacteria DNA is 1.36 millimeter, can you calculate the number of base pairs in Escherichia coli? So here note, you know all, the distance between two consecutive base pairs is 0.34 nanometers. Here the DNA length is given in millimeters. That is why here this nanometers we have to convert it into millimeters. So when we convert this 0.34 nanometers into millimeters, that is 0.34 into 10 to the power of minus 6 millimeters. Here 0.34 into 10 to the power of minus 6 millimeters distance, one base pair is located. Then the total length is 0.36 millimeter distance. In this distance, how many base pairs are located? 1.36 is divided by 0.34 10 to the power of minus 6 into 1. Then the answer is 40 lakhs base pairs. When this 40 lakhs base pairs converted into kilo base pairs, 4000 1000 base pairs because 1 kilo base pair is equal to 1000 base pairs. In this way, Escherichia coli bacteria, the length is 1.36 mm. In this length, how many base pairs are present means 4000 kilo base pairs are present. And packaging of DNA helix in prokaryotes. Prokaryotes best example, bacteria, Escherichia coli here. In this, there is no clear distinct nucleus. The DNA do not scatter throughout the cytoplasm. The DNA is negatively charged. This negatively charged DNA bind with positively charged proteins and located in particular cytoplasmic region. This region is called nucleoid. So nucleoid is a cytoplasmic region in which the DNA, single chromosome, it shows so many coils, that is called genophore, is located, and in this chromosome, chromatin organization is absent. Chromatin organization is very complex and compact arrangement that is present in eukaryotic cells only. Then the packaging of DNA helix in eukaryotes. In eukaryotes, chromatin organization is more complex, and in this what happens? Negatively charged acidic DNA. DNA is acidic in nature and negatively charged, which binds with positively charged histone proteins. These histone proteins are made up of basic amino acids like lysines and arginines. So here, this is DNA, chromatin, lengthy chromatin. This is negatively charged and acidic in nature. This chromatin here and there bind with bead-like structures. These bead-like structure, each one is called a nucleosome. This is nucleosome clear view, magnifying view. These nucleosomes, bead-like structures are made up of proteins, basic amino acids, that is lysines and arginines. In this, the histones are organized to form a unit of 8 molecule histone octamer. So here, each bead we can call it as histone octamer. This histone octamer is made up of 8 histone molecules. 2 molecules are made with H2A histone protein. The other 2 molecules are made up of H2B histone protein. The other 2 molecules are made up of H3 histone protein. And the last 2 molecules are made up of H4 type of histone proteins. Here 4 molecules are present at the basal region and the remaining 4 molecules are present at the upper region. So 8 molecules located in 2 tiers. In this way, by combining these 4 types of histone proteins, total 8 histone molecules combine and form a bead-like structure, that is histone octamer. Around this histone octamer, the DNA wrap 2 times. And here one more H1 histone. What is its role? In this wrapping around the histone octamer, the entering DNA and one cycle is completed and the outgoing DNA. Entering and outgoing DNAs are completely fixed by the H1 histone. Due to this H1 histone only, the DNA chromatin tightly wrapped around the histone octamer. Otherwise, easily it become loosened. That is the importance of the H1 histone. So, negatively charged DNA wrapped around positively charged histone proteins. Histone proteins are positively charged, DNA is negatively charged. In this way, they are very stable in their structure and form the nucleosome. Here, the nucleosome contains 200 base pairs of DNA helix. Here, whatever the chromatin, the DNA is wrapping around the histone octamer in this length, the entering and outcoming. So, in this complete circle, in this length, how many nitrogen base pairs are present around the nucleosome, around the histone octamer means 200 nitrogen base pairs are present. The number of nucleosomes connected each other and form chromatin. In this way, these nucleosomes here and there are present. So, this is nucleosome, this is nucleosome. Between these 2 nucleosomes also, DNA is present, somewhat lengthy. That DNA is known as linker DNA. So many nucleosomes are connected one by one, and in this way the chromatin formation takes place. When we observe this chromatin structure under electron microscope, that is look like a beads on a string. So beads are represent the histone octamer and the remain lengthy chain is a thread-like structure. This is DNA. And the packaging of DNA helix in eukaryotes, once again we will see the chromosome, thick condensed complex structure. This chromosome structure we can see during cell division only. Whenever the cell division completed, again the chromosome converts converted into thin thread-like structure, that is chromatin. So this is chromatin, DNA. Again, this is magnifying view. The chromatin is here and there wrapping around histone octamers, so form nucleosomes. So these are the 8 histones, 1, 2, 3, 4, 5, 6, 7, 8. These 8 histone proteins combined together and form histone octamer. Around this histone octamer, the DNA wrapped 2 times, and the entering and outcoming DNA is fixed tightly by H1 protein. In this way, it continues. The importance of non-histone chromosomal proteins, NHC. So here you can see this diagram. This is histone octamer, nucleosome. This is also histone octamer. This is also histone octamer. Between nucleosomes, the DNA, this is known as linker DNA. On this linker DNA, you can see some protein-like structures. Those are called non-histone proteins. The importance of these non-histone proteins is they make more compact, tight arrangement of chromosomes during condensed chromosome formation during cell division. And one more, euchromatin and heterochromatin. So these are the nucleosomes. Here, so many nucleosomes are present which are loosely arranged. Here the chromatin is not coiled much. But here in the heterochromatin, you can see the chromatin coiling is more and nucleosomes are tightly packed here. This region is called heterochromatin. But here the chromatin is loosely arranged and the nucleosomes are somewhat far away to each other. So this is called euchromatin. What is the importance of this euchromatin? Euchromatin is transcriptionally active. Transcriptionally active means, here I explained in the central dogma theory, the DNA by the process of transcription produce mRNA. So here this euchromatin is active to make mRNA. This euchromatin only transcriptionally active and produce mRNA. This heterochromatin never produce mRNA. In the next class, I will continue this lesson, molecular basis of inheritance, with the search of genetic material, transforming principle, biochemical characterization of transforming principle. The genetic material is DNA. The 2 marks and 4 marks question and answers of this lesson in English medium are in the description. If you like this video, please share and subscribe to my channel. Thank you.